Hello everyone, welcome to today's class. So today's class I'm going to tell you about the meiosis. What is meiosis and what all the procedures are here in meiosis. Okay, so firstly meiosis is nothing but it's a process where a single cell divides twice, okay, to produce four cells containing half the original amount of genetic information. Okay, these cells are our sex cells, that is sperms in males and eggs in females. Okay, then it is also known as reductional division. Okay, see, the parent cell which gives rise to daughter cells, parent is 2 and the daughter is half, that is n. We also simply called as 2n, which gives n, means this is a parent, okay, and the half of it means only this much, it is a daughter cell. Okay, now the meiosis also undergoes meiosis 1 and meiosis 2, that is karyogenesis 1 and karyogenesis 2. In meiosis 1, prophase, metaphase, anaphase 1, telophase 1 and cytokinesis 1, these all happens, and in prophase 1 it again undergoes 5 types, that is leptotin, zygotin, packetin, diplotin, diagenesis. Okay, so in karyogenesis 2, that is meiosis 2, also the same phases, but it's a second phase, that is prophase 2, metaphase 2, telophase 2, cytokinesis 2. Okay, same as mitosis here. Okay, and the cytogenesis 1, it is also the actual reduction division where chromosome number becomes half. Coming to the prophase 1, before that, how meiosis 1 takes place in the sense, see here, it's a interphase nucleus. You should write this diagram, and here another, inside it there is a containing of nucleolus, centriole, and the thin like structure called as chromatin material, and this membrane called as nuclear membrane. Okay, see here, I'll just explain you clearly. The chromatin material, okay, the chromatin material which you can see here is converted into chromatid here, this one, and again it's converted into chromosomes. Okay, here the C is condensation and R is decondensation. Okay, after this again the see here, after this the prophase 1 will be initiated. Okay, here first, as I told you, the prophase 1 there will be a 5. Okay, among that is first one is leptotin. Lepto in the sense thin. Okay, thin, and thin thread like structure called as leptonema. Here the diagram is this. Every diagram it will be containing a marks, as I said, this nuclear membrane, nucleolus, centriolus, and thin thread like structure. It's nothing but leptotin. In simple words, it's a thin thread like structure. Thin thread is nothing but it's a leptotin. Okay, then second one is zygotin. Zygotin in the sense, it is also known as zygonema. Zygo means always it's a pair. Nema in the sense thread. Okay, so it's a pair. Here it's a thin. The difference is thin thread and it's a pair thread. See here, it is pair thread. Here bivalent. Okay, here single threads. Okay, that's the difference between prophase 1 of leptotin and zygotin. Next, coming to this, what's this? Packetin. Third one is packetin. It's also called as pachynema. Okay, so it's pachy in the sense it's a thick. Nema in the sense thread. Always nema is a thread. There it's a single, and after that it's a double, and it's a thick. First one will be thin. It's a thick. Okay. So most important event takes place during packeting stage, that is crossing over. Here the crossing over takes place. It's a very important step. Always remember, it's a very important step. Why? Because crossing over takes place here. Then non-sister chromatids cross over each other at a point called chiasmata. Okay, it's a very important word. They may ask you for a 1 or 2 marks. Okay, exchange of genetic material takes place in the prophase 1, the variation important phase of prophase 1. Okay, very important, where this genetic materials will exchange over in packet-in 1. Okay, that is nothing but in a meiosis 1. Okay, yeah, next one is diplotin. It is also known as diplonema. Diplo in the sense two, nema in the sense thread. Always remember, nema is a thread, as I said you. And nuclear membrane and nucleolus starts disappearing here. Okay, you can see this diagram where it is nuclear membrane disappears. Here you can see, here you can see clearly your nuclear membrane is present. Okay, it's a membrane. Okay, nuclear membrane is present, but here you can't see this nuclear membrane. It's a little bit, it's a disappearing slightly, and all same. The centrioles and nucleolus, nuclear membrane is disappearing, as I said, and it's a rippling. Okay, one more is it's a presence of aster rays. Here it's a presence of aster rays. Okay, next, diakinases. Okay, diakinases is also called as termination. Okay, it's a termination phase. Here, two crossover non-sister chromatids remains attached to each other at a tip. Okay, nuclear membrane, nucleolus have disappeared completely, entirely. It's a disappeared process. Centrioles have moved opposite to poles by the spindle fiber. Okay, you can see the diagram here, and you can observe, okay, what have the changes. See here, centrioles have been moved towards opposite direction. Okay, yeah. Then coming to the metaphase 1. In metaphase 1, you can observe this diagram. See here, both chromosomes arrange themselves as an equatorial plane. As I said here, they are arranged in equatorial regions, and chromosome become more thick and prominent. Here the chromosomes are the head. Okay, best stage to study chromosomes. Chromosomes are visible more here. Okay, so you can study here. The chromosomes can be studied here. Next is anaphase 1. Okay, see in anaphase 1, you can observe this diagram. What's here? What is this? In here, it says that two chromosomes has been separated. There the chromosomes have been arranged in equatorial region, but it is here separated, and the disjunction of chromosomes takes place. That is separation. Okay, see here. I'll just slightly raise it. See. Okay, disjunction of chromosomes takes place. Two chromosomes separate from each other. Okay, and chromosomal fiber pulls it towards opposite poles. Okay, yeah. Coming to telophase 1, here the nuclear membrane, nucleolus reappears. Okay, here see, nuclear membrane is reappearing. Nuclear membrane in the sense it's a this membrane which is surrounding everything. Okay, and also spindle fiber disappears. Okay, spindle fibers is disappeared here. Chromosomes recoils to form chromatin material. Last, cytokinesis 1. Here see what happens. Usually it occurs at the same time as telophase 1, forming two haploid daughter cells. Okay, see here, this is the cytokinesis 1, and it is, see how it is separated into two daughter cells. That's the end of the cytokinesis and meiosis 1. If you like this video, it feels you very useful, so please subscribe and just stay tuned for more videos like this. Thanks for watching. Bye bye.